Okay, so today we are diving into, it's official, we have made it to the neuromuscular system. So this is kind of cool. There's other classes where we're going to spend a lot more time on how individual muscles actually contract. One of the things that all the personal training certifications want you to know is what is happening during like a muscular contraction on a cellular level. So they want you to know like what's happening on a very, very, very small level. And in order to understand how that works, we need to understand all the anatomy. So we're going to have a little bit of terminology today, just some proteins that you're going to need to memorize the names of, and like what their roles are in like a muscle contraction process. And then we're also going to look at the nervous system, which normally gets also a little bit more time. But we're going to look at the nervous system and how, it basically excites your muscular system. So your nervous system's job, is to consciously create like a thought, something that you want to do. And then it's going to travel down your nervous system and then it's going to branch off to wherever it happens to be going before it, zaps a muscle and causes that muscle to do its job, right? So we're going to be looking at those two together today. This is sort of a thinner, this lesson is a little bit thinner than how we normally go over it in some of our other classes, like Nicholas, Noel, you guys will recognize our conversation today. We'll be talking about like actin and myosin proteins, which are the proteins that make muscle contractions happen. We normally spend a little bit more time on muscles, but this class, we're going to move through it a little bit quickly because we are a little more cardio based in this class. And then also the nervous system usually gets a little more time as well. Really the big part of the nervous system we're going to spend our time talking about today is actually what's happening like chemistry wise. So we're going to look at like action potentials and how those work. So we're going to watch two videos today that are going to describe action potentials and how the electric signal gets created, and then we will talk a little bit about the muscular contractions and how they actually use action potentials as well. So we're going to basically describe to you guys how everybody in here is a human battery. We talked about generating energy, we talked about moving that energy around, and now it's all about how we actually burn up that energy to do physical work. And this will be our final lesson before what we call the midterm, which always freaks everybody out, but it's just a name for it. It's the middle of the class test. And it's only a 12-day class, so we're going to take it on day 6. So we'll be talking about what our Monday is going to look like. We're going to meet in the park in person and do a little bit of a fun cardio workout, which I'm really excited about. And then we'll have our review and so that's one of the big reasons why we want to make sure everybody's got homework 1 and homework 2 turned in today. And what's really, really cool about today is Isaiah, Kenny, and Andres, and Soroya, you guys are officially done with your very first week after today. You'll be like 1 week into the program, which is kind of cool. All right, so let's take a look here. Our objectives today, we want to describe the role of our human movement system when it comes to human movement. Like I said, we're not really going to go that deep into, you'll notice like we're not spending a ton of time talking about, sorry, I thought the wrong word was written there and I got really distracted. We're not going to spend a ton of time talking about like the anatomy of our central nervous system. Normally we do talk a lot about that. But like I said, we're going to move through that pretty quickly in this version. And then we're going to describe the role of our peripheral nervous system and how it works. And then we are going to talk a lot about what's called the neuromuscular junction. And that is where basically your nervous system is meeting your muscular system. So there's a little the little gap where they are meeting up and being connected. We will talk a lot about that because there's a lot of really cool stuff that happens there. Then we will be looking at the organization of skeletal muscle, which is really cool because skeletal muscle is the muscle that moves your skeleton. That's all the muscle that you think of when you think of muscle, right? We'll talk about like how that's made up and what the differences are there. Muscle contractions and how that works. And then we'll talk about different roles of muscle fibers. So when we talk about the nervous system, your muscles, they are able to contract thanks to what is called excitation contraction coupling. You're not going to see that term very often, but so you don't have to have it memorized or anything. But excitation contraction coupling. Let's go ahead and kind of break down what that means, right? We see excitation. That means that something is getting something else excited. Like if I were to walk into like a room full of kids and be like, "Hey, I've got a pony outside," they would all be like, "What?" And then I'll get really amped up. Or like Spider-Man's hanging out on the front door. They're like, "Huh?" And I would excite everybody, right? Well, now, like, what's going to happen is all those kids are going to rush out the door, right? And they're going to basically, run. And, I created this like chaotic event. Well, the same thing is happening in your muscles. If you've got a neuron that comes down to like a muscle fiber and it spreads an electric signal throughout that muscle fiber, your nervous system has excited your muscular system. So now that excitation is going to cause a contraction, right? Your muscle fibers are going to do what they do when they get excited, which what your muscles do is they shorten. And that's one thing to remember about today. Muscles can only pull, they can only shorten. That's the only way that the proteins go. They can relax, but they cannot push. You don't have any muscle fibers that can push. And I know that sometimes gets confusing because you're like, wait a minute. I literally did push-ups this morning. But push-ups are different than your muscles pushing. Even during a push-up, your pectoralis muscle is pulling, it's shortening, and it's pulling this bone right here closer like this. And so when it pulls like that, that causes you to move away from the ground. So yeah, there are exercises called push-push, but like, your muscles can only pull. That's the only thing they can do. Their proteins can grab each other and they can shorten from end to end. And what they can do then is they can relax. They can relax back to full length, but they cannot actively push back to full length. That is actually a really important concept because that's why we have an opposite muscle for every muscle in your body. We call that your agonist antagonist relationship, but we'll look at that. Like I said, we'll look at that later. So your excitation contraction coupling is where your nervous system excites your muscular system to contract, and it's where they're meeting, so it's a coupling. So an injury of your central nervous system or of your peripheral nervous system could alter any of this. Like let's say you were in like an accident and you broke your back, right? Well, if you sever the connection between your nervous system and your muscular system, you will lose the ability to, move any of the muscles that are attached to that part of the nervous system. Because when we look at our nervous system, I'm just going to pull up a picture of this real quick. Like I said, it's not a huge part of our conversation today, but like central versus peripheral. When you look at your nervous system, it's organized into your central system and your peripheral system. You can see there's kind of different colors here. The dark orange is your central, that's your brain and spinal cord. And your peripheral are all of the nerves that branch out from that. So, right down here at the bottom, that is your sciatic nerve. I don't know if anybody in here has ever had sciatica. If you ever get that like shooting leg pain that travels down, it feels like a pinching sensation sometimes in your low back, or it feels like a pinching sensation in your butt. So that is a really, really common disorder that a lot of people deal with. And what's happening there is basically this little nerve, your sciatic nerve, which is at the very base of your spine, and its job is to like branch out on both sides. You've got your, left and right here, is actually getting pinched between this muscle and the bone right here. And so if you pinch that nerve, the signal gets interrupted, and now it's really hard for you to get a signal down to the rest of your leg. And so, or it creates like a pain signal, and then that pain signal runs down. Or like what happens in like, you can see here, this is a herniated or bulging disc, right? And so a herniated or bulging disc can happen in any of your spinal discs. But if it the lower down it happens, the lower down your pain is going to be, because you can see that's where these other nerves are branching out. So if you get like a bulging disc in your neck, it can cause a lot of arm pain. If you get a bulging disc in your low back, it generally causes like a lot of back and lower leg pain. So, that's why, bulging discs are so bad for us. So, if you were to break this and sever that connection entirely, everything below it is not going to have a connection anymore. So that is, definitely something we don't want to have happen. But it's also one of the reasons why we look at posture so much, because, we as trainers, we're not necessarily trying to rehabilitate someone who's paralyzed, coming back from like, a literally broken back. But like we deal a lot in, herniated discs and bulging discs and sciatica and a lot of like low back pain. And they're all coming from the same situation where basically your nervous system is either being interrupted or poked or, stretched, or like all of these scenarios that it really shouldn't be in. And that's what's causing a lot of our clients' pain. So once we understand that, we can understand, okay, well, let's figure out a way to, get you into an alignment that's going to cause, that touching sensation to happen less. So looking at movement, right? Movement is nothing more than like a series of excitation contraction couplings happening in a very coordinated way throughout all of the muscles inside of your body. So I really quickly want to talk to you. I just want to show you what the action potential looks like. This video is going to, like I said, it's going to go, I think this video is a little lacking in the nervous system department. But then again, it's really, I guess we're not spending a ton of time talking about the nervous system. So I guess it does kind of make sense, but we're going to watch this second video in this crash course series, actually. This is going to show you how electric signals are generated inside of your body. How does your body use electrochemical signals? Like, I'm sure you guys have heard this a million times. If you're cramping up, what are you supposed to do? Go drink some water, eat a banana, right? Like, that's what everybody always says. Well, what's weird? Maybe you don't need a banana. Maybe you actually need a handful of table salt. Now, here's the thing.
Well, they say about the people in general say about to eat banana because of the potassium, but other things you can eat is the dates.
Yes.
Dates have a lot of potassium.
There are lots of other things. Yeah,
or
actually,
tomato.
broccoli has more potassium than bananas do, actually.
No,
yeah,
the dates have more potassium than broccoli.
Maybe. We'll have to take a look. But yeah, dates are really great. Dates are also full of sugar, which is really nice. If you're like cramping up, you're probably, in a situation where you're running out of carbs.
Yeah.
But yeah, dates are really good. So you're supposed to eat potassium, right? Well, maybe you don't need potassium. You could theoretically have enough. Maybe you actually need a handful of table salt. Now, here's the thing. We live in the Western world where we have a lot of processed food and we use sodium to package processed food. So I doubt that anybody needs the salt part of this equation. But it's possible. I mean, it's possible that you might need some sodium rather than potassium. But generally, it's potassium that people are lacking in. I'd say 99.9% of times. But why sodium and why potassium? Well, the reason why is because those are your electrolytes. So when you take like a Gatorade, if you actually look at Gatorade, ingredients list here, we're going to take a look at what Gatorade actually is, because it's supposed to be an anti-cramping agent, right? And it is. Like, for the record, it actually is. So we look at it, water, which is also going to help with cramping because that's going to make it easier for your body to move materials around. Sugar, which will give you energy, which is important during sports events. Dextrose, which is, by the way, more sugar. Citric acid to give it some flavor, artificial flavors. Salt, sodium citrate, which is more salt. Monopotassium phosphate, so that is, your potassium. And then a little bit of food starch, a little bit of an alcohol, and a little bit of blue one to give it a weird color because,
What about to drink something electrolytes or prepare something the water with salt only, without all these kind of things? Because I think this is too much chemical.
What do you mean? You mean like an electrolyte powder?
Well, I when you are deshydrated, the doctor sends you something like you drink the water with salt and give you and help you to
Yeah, so you actually
recover. But without to drink this kind of thing, well, I don't like it, and I see they have a lot of chemicals. I hated that.
So here's the thing: I am not a huge Gatorade fan, but statistically, like Gatorade was actually, it was actually invented at which Florida school is it? No, you probably know this, right? Or no, wait, I'm sorry, that's the wrong East. Sorry, I'm getting my states backwards. Um, but I can't remember which Florida college it is, but the one with the Gators. It's either Florida U or the other one, or Florida State. But anyway, they invented Gatorade, to help their athletes who are cramping up. And originally it was just they put this main one here, sodium citrate, this one right here, in water, and they found that it was a really good way to make sure that their athletes got hydrated, and then it was a really good way to make sure that they had sufficient sodium inside of their cells, which would make it easier for them to create these action potentials that we're talking about. And then, they were like, hey, we could probably make $1 million off this. And so they threw in food coloring to give it, a weird look. And then they added a lot of sugar to make it taste better. And now we have what we have now. But yeah, Soraya, you don't have to take Gatorade. I don't personally like Gatorade. I never drank it as a kid. So like, I missed the train, all these people that I play sports with, they all love it because they all grew up with it and stuff. And yeah, if you want to do like a much more natural, if you want to do a much more like natural way to get a lot of like electrolytes in, you can do like coconut water. Coconut water is packed full of electrolytes. Actually, it's got.
Oh yeah, but not this kind, because this is as it says the same like the natural coconut. They put too much things. They don't.
I mean, it's coconut water, and then it's my banner's in the way. Hold on, I can move it. It's coconut water, and then less than 1% of natural fruit sugar, and they do add vitamin C. So they're adding a little bit of vitamin C powder, but that's pretty natural. It's not going to get much more natural than that.
Well, I tried most of them to try because I like the coconut water, but doesn't taste the same.
Oh, I don't doubt that there's definitely some, souped up chemical-y versions of coconut water out there. I definitely don't doubt it, but natural coconut water has a lot of sodium and a lot of potassium in it, which means that it has a lot of natural electrolytes. And so that's what I want you guys to really know today. When you hear the word electrolytes,
when it comes to getting healthy,
your electrolytes are sodium and potassium. And they are electrolytes because they are what is going to generate electricity inside of your cells. Your body is going to use those to generate action potentials. Okay. And then Soraya, we'll talk a little bit more later about when we get to nutrition, we will cover like natural foods. And we'll talk a little bit about like which chemicals are bad for you and which chemicals you really don't need to be afraid of. There's actually plenty of chemicals in your food that are super, super fine. And they don't have any negative effects on you. But when they are man-made chemicals, that's where we start to get in trouble. But you got to remember, water is a chemical. The definition of chemical is two atoms combined together. And so
yeah, I know, but I refer more about what the people is using in the products.
Right. Well, we'll talk about that more later, for sure. When we get to nutrition, that'll be a big part of our conversation.
Okay.
And you'll hear me rant about artificial sweeteners and why they're so bad for us. But let's go ahead and take a look at action potentials here. So this is, like I said, this is technically the second half of the Crash Course Nervous System video, the Crash Course Nervous System talk, but this is the part that we need. So
what if everything you did, thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses. Like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we've got to first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy. But it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membrane. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge.
So I just want to pause it real quick. So that actually is sometimes a little bit of a weird concept, because you're like, wait, sodium and potassium are both positive, but there is way more sodium outside of a cell compared to inside of the cell. So the difference between the two, it's like, sodium might be worth like 3 and potassium might be worth like 1. And so, there's just more, positive out here than there is out here. So you are in a negative place when you're resting, and you have the potential to become positive and create an action potential at any time simply by flushing those concentrations and flipping it. And like he was mentioning just a little bit there briefly, You know, if you have a positive charge over here and you have a negative charge over here, nature wants to cancel those out. Nature does not like having a lot of stuff on one side and a lot of other stuff on the other. Nature wants everything to be really, really, really even. And so if you are separating that with your cell membranes. One of the reasons why we need to make sure we have fat in our diet, especially essential fatty acids, they are what give you cell membranes. And so it's really important that you have those. And so, like, you know, fish oil is very good for you. But like when you separate those potentials, right? Your body wants to be able to switch the concentrations. And that's what we're going to get into here. He's going to talk about how there's these little pumps that will allow you to push stuff in one direction, and then stuff can leak out in the opposite direction. And now you have flipped your charge. Just like a battery, you turned the neuron on. So.
Neuron has a negative membrane potential like this. Ions outside, then there are potassium ions inside. The cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts, but some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions.
So again, sorry, I want to pause that. So those different types of like channels, right? Those different ones he mentioned, you've got ligand gated, you've got mechanically gated, and you've got electrochemically gated, right? So, oh, whoa, I lose 2 of you guys at once. Hey, sorry, Kenny, welcome back. I can never tell if that's my internet kicking you guys off or what, but so when we look at these different gates, right, that are allowing sodium and potassium, there's different things that will make them happen. So for instance, on your muscles, when your nervous system zaps your muscles, there's electricity running through your muscles, and when electricity runs across it, that activates the electrochemical gated ion channels, so they open up. That's also why when you ever like shock yourself, you'll contract sometimes. That's exactly why, because like basically a bunch of electricity is running through you and it causes your muscles to contract. You can take dead tissue, and as long as it has like a little bit of like leftover ATP and stuff inside of it, you can zap it and it will jump. You've also got ligand gated, which is where like if a neurotransmitter or if a hormone binds to a specific receptor, that will also cause it to get excited. So that is how adrenaline and other materials actually amp up certain parts of your body. Even like serotonin or dopamine or oxytocin, which are all really positive, happy-go-lucky hormones. There's one literally, oxytocin, by the way, they call it the cuddle hormone. And your body will make it when you give someone a hug. And it makes this, like, you will generate oxytocin. Well, when oxytocin binds to certain parts of your brain, it causes this feeling, like this really positive, good feeling. And that's sort of how we like experience like affection. And so that causes that type of action. And then there are also, and this is probably the coolest one, mechanically gated ions. And they actually, when a tissue is stretched, that causes them to open. Go back to our digestive system conversation. Remember how food gets broken down, and then at some point it runs into our small intestine, but it can't get into the small intestine because that sphincter is closed, right? Well, once it is soft enough and the food is broken down enough, that's what trips those mechanically gated channels. And that's what causes the muscular contraction to open that up. That's also what, when you are actually stretching and all of a sudden your body is like, that's it, stop, and you're stretching, you're like, oh, and you feel that pain sensation. You are getting to a place where you are stretching certain sensory receptors inside of your muscles that are causing you to not want to stretch any further. And it's because the signal that you're getting, the pain that you're feeling, is being generated by these mechanically gated channels. So different channels do different things, and they are all, every one of them has an effect during our personal, during our sessions. Whether you're stretching a client or, having them jump explosively, or if they're just digesting their meal after they leave the gym. All right, that'll be my last interruption.
Running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed, and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. So kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean. Once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time, the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, Your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of
actually, hold on, I gotta
batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale. The script was edited by Blake T. Pestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot. I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's good, and then forever, it can be helpful to people. But younger people, not so much. And so we are creating Crash Course Kids. Hosted by Sabrina Cruz from Nerdy and Quirky, Crash Course Kids will start out focusing on 5th grade science, but we'll keep expanding to other topics as the channel grows. Sabrina will be talking about food chains and gravity and how the sun works and how plants eat and why flamingos are pink and many other topics. Oh, and another note, teachers, you can rest assured that we've got you covered. There will be info about the standards we've used to make sure that we're doing our very best to help you out. So if you are a teacher, or you know a teacher, or you know a child, or you know someone who has a child, or you've ever seen a child, you can tell them to go to youtube.com/crashcoursekids and subscribe, and you can go do that as well if you would find that kind of content useful or interesting.
Okay, there we go. Sorry, I always try to show the entire videos because, like, you know, we're using these for free. So I just try to make sure we get all the credits in every time. Okay, so that is obviously, there's a lot of chemistry in what we just looked at, right? Like there's a lot of like moving sodium and potassium and things like that. But again, that's generating these action potentials that are responsible for literally everything that we do. And so when we talk about the neuromuscular system, we have to understand how action potentials are created, because one of the things he mentioned in the video is that what's interesting about this is, you know, how is it that I can, you know, go pick up a really heavy barbell, you know, and you know, for as much strength as possible, you know, rip that heavy metal off of the ground and then set it back down. And then I can use the exact same muscles that I used to do that to pick up like a puppy, right? How can I use the same thing without like grabbing that puppy and then going like, you know, throwing it through which would be terrible, right? Like, how do I make sure that I don't do that? Well, there are ways that we regulate those signals, but here's the thing. The signals are always the same strength. Like he mentioned, it was 70 millivolts and negative 55 millivolts, right? So if the signal is always the same, how do I have like a strong contraction versus a weak contraction? Well, the speed of those signals is actually what we can regulate. If I send one signal after the other and it looks something kind of like this, that's not really that strong of a signal. And that's maybe me, you know, grabbing a door and opening it, right? But let's say I'm doing like a medicine ball slam and I'm explosively contracting my muscles as hard as I can. That signal is going to sound something more like this, right? It's going to be like bah, 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 much, much, much faster. That is a powerful contraction versus a very weak contraction. And depending on how much of how many muscle fibers you have, how many muscle cells there are, and how much like action potential your muscle can generate, that will determine the difference in your contractions. So, you know, part of what we do as trainers is we are trying to build muscle in our clients. What that does is that gives them more muscle cells, that gives them more muscle, you know, more muscle fibers. By having more muscle fibers, they are able to create a better action potential in more at the same time, then all of those proteins can work together. But if I want to be really strong and not just big, I need to make sure that I am also training my nervous system to get all of those coordinated and contracting at the same time. This is one of the reasons why, for those of you who have done maximum strength lifting, if you've ever been in like athletics where you have done, you know, like 1 to 5 repetitions. Like if you've ever done like a push press, for instance, right? You jerk that up overhead and you bring it back down, and boom, like 1, 2, and then like maybe one more, like 3, and then you set it down. You rested for like 5 minutes after 3 reps, which sounds like, you know, a crazy work to rest ratio. But the reason you take all of that time is not only to restore all of your energy so that you can work really hard in your next set, it's also to ensure that you give your neurons all time to relax. Because after doing a really heavy stimulus like that, your neurons are like, what are we doing? Are we fighting a bear? Let's do this. You know, like they're all turned on and they're amped up. And so now you might have a hard time coordinating them and getting them to fire in sync. So by giving them lots of time to relax, you're giving them time to be able to work together. So you are actually taking long rest periods, not so much for your muscular system, but partially for your nervous system. So that's getting them synced up. And then the other part of it is like, well, what if I can get more neurons getting, you know, the attention of more muscle fibers? So now I get muscle fiber recruitment. Well, that's also going to result in more strength. More muscle fibers, obviously, more proteins causing more contractions. And then, so now I've got muscle fiber recruitment, muscle fiber synchronization, and then their firing rate, how quickly they can create those signals. If they can create them over and over and over again, that's an even more powerful contraction. So that's why we got to talk about our nervous system whenever we talk about the muscular system. So like I said, it's going to be under the control of these action potentials, right? And so a lot of this is under control of like your brain. There's this area called your precentral gyrus, which is located in the front of like this, what's called the sulcus of your brain. And that's like these little lobes right back here in the back area. And that will be attached to what is called your motor cortex. The motor cortex is really the head honcho here. That's like, you know, the air traffic controller for sending signals to wherever it is you are trying to move. You know, next time you miss something, like when someone throws you something and you know, reach your hands out, it hits you or it just goes past you and you miss it, you can blame your primary motor cortex. And so the architecture here is just a little bit different compared to the rest of your nervous system. It really is designed to fire very quickly and to be able to like get immediately into your muscular system as fast as possible. And so, you know, again, your brain is at the center of our conversation whenever we are talking about muscular movement. There's always a connection. That's why, you know, a lot of times we don't just call it muscular, we call it neuromuscular, because we are describing, you know, how our nervous system is coordinating our muscular system. So obviously when it comes to muscles, right? Muscles and neurons both, this takes a lot of energy. Remember, like sodium and potassium, if they had their way, they would just bind up together and cancel each other's charge out and just, you know, be totally neutral, right? Nature would just be like, you there, boom, canceled out, right? But luckily, we have all of these cell membranes that are separating those things. And by separating those things, that creates, like you mentioned in the video, potential, right? It creates the ability for us to be able to move this stuff around. And so now how do we move stuff against like, you know, the way nature wants to move it? Well, we burn up a little bit of energy, and that is where our mitochondria come back. Our conversation, you know, again, it all comes back down to talking about energy production, right? So your mitochondria, which are distributed within your muscles and your neurons, right? It's saying muscles here, but they're distributed everywhere. Are going to be burning up the energy that pushes these things against their concentrations. You actually saw it happen in, it was happening in the video as you're watching it. But if you look at like ATP and action potentials, I need to see that little protein. Come on now. Help me out, Google. All right, let's try sodium, potassium, pump, ATP. Let's try that. There we go. Okay. So take a look right here. This is kind of what those pumps look like, right? You can see there's a little bit of area for sodium and there's a little area for potassium, right? Now, you see this like, you can see actually sodium is going to want to move this way and bind to this. So basically this little pump, it kind of rests like that. And then, you know, sodium can rush in and then it will move like that. And that will allow that sodium to get outside of the cell or inside, you know, it depends on what direction like it's being pumped, right? And so it's moving sodium and potassium concentrations there. And you'll notice, it's kind of funny because they're moving it to the other side in this picture here, but there's a little molecule of ATP and they got it, you know, drawn like a little lightning bolt here. So there's the energy, right? So remember how I mentioned that ATP, it's a molecule of adenosine and then 3 little phosphates. You know, it looks like this, right? Here's your adenosine right here, and then there's your 3 phosphates. So when you kick that 3rd phosphate off, that's how energy is released. And you're left with ADP, you get 2 phosphates, right? Diphosphate. So you can see here, there's a little bit of ATP, and it will leave one of its phosphates behind. It breaks it off, and that is what causes a ton of energy to be released. And now this little protein inside of your cell will change its shape. Well, I guess actually it's going this way. It will change its shape, and then it will release that phosphate so that a new molecule of ATP can bind to it. Okay, so that's what's happening. A little ATP comes in, a little bit of energy, it breaks off, it pushes sodium or potassium in whatever concentration it needs to go, and then, you know, it's able to kind of move back to its original shape once the new ATP molecule binds to it. And that's happening over and over and over and over and over again, which is why it takes so much energy in order to move us around. This is why we got to make sure we have plenty of glucose and fats and calories, really, at the end of the day, in our diet. We got to have enough energy to be able to do all this. So your mitochondria are going to be distributed. They're there to produce this energy. And during a contraction, each myosin will break down some ATP, which will obviously release a bunch of energy, and then that will create that muscular contraction, and it will also create like the action potential in a neuron. So, you know, this is happening in both of these things right here, right? This is either going to trigger the ATP action inside of a muscle cell, or it's going to be, you know, every single part as it's moving down your neurons. Both of those are using the same situation. They're both using action potentials, okay? And then here's the different parts of your brain that do that, by the way. I'm not going to spend a ton of time on this, but there's your there's your motor.
Teacher, I have a question. So
yeah, what's up?
ADP wanna be the potential?
Mm-hmm. No, not wait, ADP?
ADP.
Yeah, so ADP is a burnt up ATP. Your body will take that ADP and it will add a phosphate back to it so that it will have energy again. But it takes time in order to do that. So your body will your body can't really use, it doesn't use ADP very effectively for energy. There's not as much energy trapped within it. So your body doesn't prefer to use that. So instead, anytime you're left with ADP, your body will try to reconstitute it to ATP. And actually, it uses an enzyme called creatine to do that. For any of you who have taken creatine supplements, that's actually why you take it. We make it naturally, but you can boost your levels through supplementation. Does that answer your question?
Yeah, thank you.
Yeah. It can use ADP, by the way. It can even use AMP, which is 1-phosphate, which you do when you do exceptionally like marathon training. When you do like super long runs, sometimes you'll like really break down those other ones. But that is super low intensity energy compared to ATP.
Hey, I got a question.
Yeah, what's up, Noel?
When it comes to the neural system, right? Don't the if you have bad allergies, can it play a big part of that?
Not really.
Oh, okay.
No, your nervous system, allergies are a response of your immune system. I mean, I guess that there's definitely like there's a connection there, like you might get like your nervous system might send something out in response to your immune system, but it's all triggered by your immune system. So allergies are part of that. Like everybody is allergic to mosquitoes. That's technically an allergy. Whenever you get that little mosquito bump, that's technically an allergic reaction. Mosquitoes don't have any like poison that they're putting in you. They just have a little like enzyme that is supposed to like, it's basically like, you get a shot of like Novocaine or something and it keeps you from feeling it. They have that on their nose. And so when they puncture you, you don't really feel it. That's why like you'll look down and be like, you mother, like, and then you just notice it later. So the thing is that when it's left behind, your immune system is like, what is that stuff? And so it freaks out and it sends a bunch of like histamines to the area. And that causes it to like swell up. So it's an allergic reaction there. That's your immune system, not your nervous system. That's my best understanding by the way, I am no expert on the immune system. We are in the wrong part of biology for me. If it's not muscles, I don't know it. So your cerebrum, the large part of your brain, and it divides down. And again, it's going to regulate your motor system. Then you get your cerebellum, which is going to help you process information. So it's using both of that. Your medulla oblongata, that is actually, your medulla oblongata is a lot of your automatic functions, like all the stuff that you really aren't thinking consciously about. So even like when you're stretching and that pain sensation shows up, a little bit of that's happening in your medulla. But don't worry about the different parts of the brain. That's something that you will definitely never talk about again. But what we do want to talk about is, that's all very much our central nervous system, right? That's our brain and our spinal cord. But the peripheral part of our nervous system are all the branches that are going to branch out. You can actually see that right here. This is like a drawing that's been cut off. But you can see your C5, C6, C7, C8, and then it gets into your T1. That is the top of your neck heading down, and then it's the first part of your thoracic spine right here. This is your T1, okay? So there is your cervical vertebrae, that's your neck, and then your thoracic vertebrae is the very top of your upper back, right? And you'll notice there are little branches that branch off from every one of these, and then they'll branch down smaller and smaller and smaller and smaller, just like veins or capillaries will. And they get smaller and smaller until they get to your tissues, and then they cover your muscular tissues in order to zap them and make them do their stuff. So this is your peripheral nervous system. It's all the nerves that have branched out. So I want you to think of the central nervous system. Think central is close to your midline, right? Brain and spinal cord. Peripheral nervous system is along the periphery, right? It's around the outside. And that's all the nerves that branch out as far as they're going. If you know anybody who has like diabetes, they might be struggling with what's called neuropathy. Anybody who has ever had neuropathy, or like, sometimes just happens as we get older due to other conditions. Neuropathy is where essentially you'll have like a dead sensation. You won't be able to feel certain parts of your fingertips or certain parts of your feet, legs, whatever. It is basically a loss of, it's nerve damage that results in a loss of sensation. Your peripheral nervous system is being interrupted. There's no signal. There's plenty of signal going out, but there might not be a signal coming back. And that's not very good because, like, you know, that's like me being like, you know, like right now, Andres, you don't have your camera on. If I said, hey, I want you to do a bicep curl for me, I have no way to know if you actually did it or not, right? That's kind of what nerve damage, you don't have to, there we go, I love that. You don't actually have to do it, but like, yeah, that's, without that me happening, I would just ask over and over and over and over and over again, you know, because I don't get any information back. And that's why neuropathy is so dangerous. That's also what's happening when you sit on your leg too long and your leg falls asleep. It's not that you're cutting off blood flow to your leg, you are cutting off your nervous system's connection. So your nervous system starts screaming, trying to get your leg's attention. And then you shift your position and you start to feel all that screaming and you feel all those little pings. That is literally, you know, an actual, like a ping is a really good way to describe it. It's literally your nervous system saying like, hey, are we still connected down there? You still there? Right? And then your leg's like, yeah, I'm still here. And you feel all of it. So, all right, so there we go. Now we're done talking about the nervous system. We will leave the nervous system alone, and we'll move into the muscular system. So inside of your muscular system, this is where the conversation gets kind of fun. Inside of your muscular system, you have all of these what are called myofilaments, okay? Myofilaments is a specialized type of protein. They are able to bind to each other, and that's actually what creates muscular contractions. So if I pull up a picture here, and we look at myofilaments, you can actually see they're arranged like this. They form these little cross bridges. And you can see like there's just a little bit of a connection here. They're just barely crossing over each other. And what happens is this reaches up and grabs this, right? So this right here is your thick filament. This is called myosin. And these thin filaments here, these are called actin. And what happens is myosin reaches up and grabs actin, and it's sort of like passing it hand over fist, kind of like if you were passing something to me, if you grabbed a rope and you were pulling on it and then you were like, don't let it get any slack, and then I were to grab it behind you and keep the slack, and then somebody else grabbed it behind you, you know, if we were all pulling on a rope like this, what would happen is, I'm actually just going to copy this picture here. Where's paint? I need to put paint in my shortcuts. You know, what would happen is like this cross bridge here would move this way. And it's going to travel like that. And that's happening in our other side as well. And that's going to move this way. And so now this muscle cell is going from this really lengthened position up top to this shortened position down below. And now you can see like there's really not much more room to move here, right? And they're already at the end, there's not really much room to move over here either. So that's this is a fully contracted muscle cell. And that's what we're looking at. That's what's happening in every one of your muscle cells. So if that happens in this muscle cell, and you can actually see right here, that's the muscle cell, this is one individual cell, and then it's attached to this one, and it's attached to this one, right? If that happens in all of your muscle cells simultaneously, you're going to have a really powerful contraction. But what happens if it only happens in half of your muscle cells? Well, then you're going to have a weaker contraction. And that's another way that our body regulates strong contractions versus weak contractions. So based on everything that I've said today, you should be starting to put it together where it's like, okay, so what strength really is at the end of the day, like the difference between strong and weak is how often I can create these action potentials, how much of my muscle fibers I can recruit simultaneously, and then finally, how many, actual muscle fibers I've got. So you want to get strong, you need to be able to have lots of muscle fibers. You need to learn how to recruit them as fast as possible, and which is a nervous system thing. And you need to be able to create enough energy in order to be able to do that as explosively as possible. And that is going to translate to low repetitions for a very high intensity of your maximum effort and lots of rest to make sure that you have enough energy to do this every time. So actually, Noel, you didn't even realize it when you asked this morning, but you set me up perfectly for this moment. Which is better, sprinting or long distance running? Well, it depends. Are you trying to get your muscle fibers to contract for long periods of time? Then I would say a long, slow jog is best. But are you trying to get strong? Then I would say sprinting with long rest periods is best. Ha! That was handy. All right, let's go ahead and watch our last crash course video here for the day. And so here it is. Here's the here is the Romeo and Juliet
Okay.
analogy.
Mhm.
I like this one. This is one of my favorite videos. He moves really fast in this one, but I really like this one. This has got the best animations in any crash course video. This is my favorite one.
Romeo and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation, and poets and emo rockers love them for it. But you know where else you can find a nice hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of
Turn it up a little bit.
there, in fact.
What's that?
Can you turn it up a little bit?
Actually, I don't think I can. I don't. It's kind of default on my side. Here, I'll tell you what. I'll try. If I turn, I don't think turning my volume up will help, but I'm gonna max it out. If it helps, let me know.
Situation, but deep down in the cells of your muscles, the hot protein action between
Does that work?
Good. Good. It's good.
Okay. All right. Cool. I did not know that worked that way. Here we go.
actin and myosin is actually literally causing all of your motions, all of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy or movement simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract. You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach and airways and blood vessels, where it involuntarily and very usefully pushes fluid and other material around by contracting and relaxing over and over. Your heart is so important that it gets its very own muscle tissue type, cardiac muscle, which looks striped or striated and also functions involuntarily to keep your blood pumping without you having to think about it. But when you hear the word muscle, you're probably thinking of the kind that you see on Chris Evans when he first walks out of that machine in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii or vastus lateralis or gluteus maximus, is technically its own organ made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, each one is rigged up with its own personal nerve to stimulate contraction and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squished together to form muscle fibers, which are your actual muscle cells. cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise you need a little help. That's why every muscle contains a few different kinds of supportive sheaths of connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1, proteins like to change shape when stuff binds to them, and 2, changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. All right, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I'd prefer cash and nachos, but whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now, I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go. At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium. And remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle. That's the sliding part of the sliding filament model. Now, with its energy spent, that little head has no use for the ADP and the phosphate, so they unbind with it because, remember rule number 2, changing shape encourages proteins to bind or unbind with stuff. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin in a tear-jerking scene like some microscopic recreation of the finale from Titanic. But fear not, this epic is not quite over, because this is when the myosin breaks down its new molecule of ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum, so they start grabbing the calcium that's floating around, causing calcium to unbind from the troponin. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back into place. It's a circle, or potentially a big Hollywood franchise with lots and lots of sequels. It keeps repeating itself many, many times every moment while I sit here and talk, and while you sit there and eat and text and take notes, the whole drama replaying itself over and over. Kind of like you'll have to play this video over and over again to get all the little steps of the sliding filament model straightened out. But hey, some stories get better the more you hear them. If you do watch this one again, you will relearn that your smooth cardiac and skeletal muscles create movement by contracting and releasing in a process called the sliding filament model. You'd also relearn that your skeletal muscles are constructed like a rope made of bundles of protein fibers, and that the smallest strands are your actin and myosin myofilaments. It's their use of calcium and ATP that causes the binding and unbinding that makes sarcomeres contract and relax. Special thanks to our headmaster of learning, Thomas Frank, for his support of Crash Course and free education. And thank you to all of our Patreon patrons who help make Crash Course possible through their monthly contributions. If you like Crash Course and want to help us keep making great new videos like this one, you can check out patreon.com/crashcourse. Crash Course is filmed in the Dr. Cheryl C. Kinney Crash Course studio. This episode was written by Kathleen Yell, edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. Our director is Nicholas Jenkins. The editor and script supervisor is Nicole Sweeney. Our sound designer is Michael Aranda, and the graphics team is Thought Cafe.
Okay, so let's go over that slowly. So looking at what's happening, we got to first talk about like the anatomy of everything. So here's some of the key vocabulary you're going to look at today. So inside of a muscle, you are going to, and by the way, like one vocabulary word that we're missing here, you're going to hear me use the word muscle fiber a lot. That's a big fancy way of saying muscle cell. So when I say muscle fiber, I just mean it's an individual muscle cell, just like you have nerve cells or skin cells or whatever. So inside of a muscle cell, you are going to find myofilaments, okay? Myofilaments are these specialized proteins that are able to bind to each other and create a contraction. You will see also the term myofibrils sometimes. It's part of that's composed, it's got actin and myosin. Yeah, myofibrils, myofilaments. They are slightly different, but for our purposes, they're the same. And on there, you are going to see a thin strand, which is called actin. And you're going to see a thick strand, which is called myosin. Okay, so going back here, you can see this little thin strand here. This is the actin strand, right? This is actin. And actin, when you zoom in on it, it has binding sites. Actually, you know what I can do? I can totally steal a picture. Let's see. Oh, that's perfect. Copy image. And paste. We're going to put you. Oh, they did it in with a black background. That's fine. We're going to put you over here. All right. So this is what actin actually looks like when you zoom in, right? I guess I got to use a different color arrow here. So this is your actin strand right here. Okay. And you can see when you zoom in on it like crazy, right? It has these little binding sites. See all these little tiny blue dots, right? Those are all binding sites that these really large proteins, which are your myosin, more space, that your myosin wants to bind to. So myosin have these little heads on them that can grab actin actin strands, okay? They're not going to grab, not the red ones. We'll talk about what those are in just a second, but they're going to grab any of the little blue bits there, okay? So that is actin, and then this here is your myosin. Okay, so actin is going to get grabbed by myosin, right? We said like these are like, you know, high schoolers at a dance. They really want to get up in each other's business. So they have these little club-like heads that will grab their binding sites. Now, because these proteins are so attracted to each other, they want to bind all the time. They really want to reach up and grab. And you know, we don't want that to happen when we have our muscles relaxed. So you can actually see, you can't see it on the left picture here, but if you look at the right picture, you see this green little strand right here, and then you see this little little pink strand right here. Those are what we call regulatory proteins. Those are troponin and tropomyosin. Their job is to block the binding sites, okay? So their job is to like have this little rope strand here that keeps them from binding. And you'll see they've got a little like binding site of their own, right? And that's going to be for them to grab a molecule of calcium, and that will move them out of the way. So that is troponin. And then the strand that it's attached to, right, that is also tropomyosin. So they're there to block the binding sites. Their job is to keep that binding action from happening, but they can be paid off. You can actually get them out of the way if you just flush a little bit of calcium into the area. So if you flush a little bit of calcium into this area where all of those proteins are at, troponin and tropomyosin will move out of the way. That will expose the binding sites. And now myosin is like, it is on, and it reaches up and grabs the actin and it pulls like this. Meanwhile, that's happening again and again and again, and your muscle cell is going to shorten. That's also why your muscle cells all bunch up whenever you perform a contraction. You know, it goes from being in this long position here to this kind of bunched up position like that, and it causes like a little bulge, right? So that is the sliding action. And that term right there is one that you are going to hear all the time. We call that the sliding filament theory or the sliding filament model. That's what's happening here. These proteins are sliding, like we said earlier, these guys are sliding this way, and these guys are sliding this way. And so the whole muscle cell is shortening like this. Now, there is another term here that's actually not in our notes today that you're going to hear later in other classes. The area where this is happening, we call that the sarcomere. You'll hear NASM, they'll define it as it is the functional unit of the muscular system. And basically, it's where the sliding action occurs. The sarcomere isn't like a physical thing. Like, these are proteins, right? Like, you could grab a protein. Like, they're physical things that are in this world. Sarcomere is just a term for like an area of town, you know? It's the downtown of a muscle cell, right? And that's where all this sliding action occurs. But that sliding filament theory is where the sliding action inside of muscle cells is occurring. And that is why our muscle cells are able to do what they do. Now, like I said, they're under the control of our nervous system. So, you know, they can't just break ATP down willy-nilly. They have to be told to do it. And that is when the sliding filament will occur. So an ATP molecule will be broken down. It will release a bunch of energy. That energy will then be used to trigger the sliding part of the sliding filament theory, right? Or the sliding filament model. So how does that actually happen? Like, what triggers this event? Well, that's where motor neurons come in, right? So you've got a lot of different types of, like, you know, neurons throughout your body. You've got sensory neurons, which, you know, he mentioned earlier in the video, when a spider brushes up against your skin and you feel it, right? That's where like maybe a certain stimulus triggers a neuron to go, whoa, what was that? And then you realize that it's a spider. You got sensory neurons, you've got integrative neurons, which are there to like basically just interpret information from the last time that something like this happened. But the big one that we're talking about here are motor neurons. And motor neurons are the neurons that are specifically heading away from your brain and zapping your muscles. So they're not going back towards your brain. These are the ones that are going to your muscles themselves from your brain. And motor neurons create that contraction. They experience that excitation contraction coupling that we mentioned earlier. It's where motor neurons innervate or excite your muscle cells, right? Your muscle fibers. So that is what we're looking at there. Now, and you'll sometimes you'll hear this also referred to as motor nerve, but very rarely. And that's a term that, like I said, motor neurons are neurons in the nervous system that connect to produce your muscle actions. They create this happening. So you can kind of see that all happening right here, right? You've got an overall muscle, which if we zoom in is just a bundle of bundles, right? Like here's like an individual muscle cell. And then we have like that muscle cell right here being bound up with a whole bunch of other muscle cells. And then all those muscle cells are being bound up with more, bundles. And then those bundles are all wrapped in one big sheath. So that's what's kind of fun about muscles. They're kind of like a rubber band ball, like it's like an individual muscle cell. And then you get like a bunch of those together and wrap them all up together. And then you've done that with a bunch of other ones. You wrap those all together. And then that actually makes up your overall muscle. That's what's called a fascicle, by the way. So we can see here, this is the sarcomere, and then this is a different sarcomere, and that's a different sarcomere, and it's the area where this little crossing is happening. And then there's your little proteins there. So again, little individual myofilaments, an individual muscle cell. That muscle cell is bound up with a whole bunch of other muscle cells. Then they're bound up together, and then, one with like, we'll say 50 or 60, and then that 50 or 60 is bound to another 50 or 60 and another 50 or 60, and it'll eventually becomes your whole muscle, and you got thousands of cells all firing at once. And there's different layers to all of this. We're not doing layers today, are we? Yeah, I didn't think so. There's different layers that are allowing all this to happen. You've got these different layers of skeletal muscle. You've got these are going to be similar to our heart terms that we used yesterday, talking about the layers of the heart. You've got an epimysium on the outside, a paramysium, which is like in between, but still outside. And then the endomysium, which is the innermost layer. But don't worry about those for now. You're not going to see those terms come up, actually, probably ever again. So how is that chemical reaction actually taking place? Well, here it is, right? Here is what is called the neuromuscular junction. This is a big reason why our nervous system can interact. So take a look right here, guys. See all of these little mitochondria? The end of this neuron is creating a lot of energy, right? It needs to be able to create energy all the time. And then see here, this is your muscle. So this top part is a neuron. And then this bottom part is an actual, it's grabbing a muscle cell, right? And so that's also got its own mitochondria. And so that's also producing energy. And then there's a little gap in between the two. That junction, that little gap right there, that is your neuromuscular junction. And what you're seeing is all of these little tiny dots right there. Those are called neurotransmitters. And so neurotransmitters are actually what create this contraction and allow it to happen. So the neurotransmitter that we're going to use is what's called acetylcholine, okay? So actually, muscular contractions, when the nervous system interacts with your muscular system to create that excitation contraction coupling, the nervous system will trigger your muscle contractions using neurotransmitters. So neurotransmitters are chemicals that trigger physiologic events. If you had a, we're gonna keep talking about donuts all week. If you had a donut right now and you were to like grab like a bite of it, right? The sugar that's in that donut would excite your taste buds, and your taste buds would go, wow, that is a lot of sugar. Holy crap, that's bad for me. No, that's not what your brain's thinking. Your brain's thinking, wow, what a readily usable source of energy. That's why sugars are so addictive, by the way. Your brain doesn't know any better. It thinks that you're a cave person, and it thinks that sugar is rare. And so when you bring that like sugar in, what happens is your taste buds fire like crazy. Well, your taste buds use a series of chemical reactions. And then you might get this like pleasant sensation in the back of your brain where your brain goes, yeah, we like sugar. Give me some more of that. That's going to keep me alive. Your brain does not know that it will also give you diabetes. But it will create neurotransmitters, which will make you feel really good. And that's how people get addicted to sugar. If you were a smoker and you bring in like nicotine, your brain will make neurotransmitters that go, whatever the heck that was awesome, right? And that is how addictions are. That's why they're so easy to get. It's incredibly easy to get addicted to things because your brain is wired to fire up these neurons and they create these neurotransmitters. That's what's interesting about habits. And we are going to talk all about habits next mod. But the big thing to remember, I'm just going to touch on this right now. If you guys ever want to talk about habits and the psychology and physiology of habits, I love talking about that. So, you know, we can always talk about it after class or anything. But it is interesting. Habits are not concepts. A lot of people, it's like, oh yeah, I get it. I have a addiction to video games or whatever. And it's like, you don't just have an addiction to something. You have a physically altered, tangible, we can hold it in our hands, physical change in your brain. That's what addictions are. That's why habits are so easy to form. They're very, very, very easy to form. And the habits that are worse for us, the reason they're so easy to form is because generally they're just really, really, really strong impulses. And rewriting those takes time and effort. But if you think of them as physical things, well, do you believe that you can gain muscle? Do you believe that you can lose weight? Do you believe that like you can change the shape of your body through time and effort? I mean, I don't think anybody would be in this class right now if they didn't believe that, right? That's literally our job. Well, we can also physically change the shape of our brains as well in really positive ways and overcome our habits. So anyway, I'm sorry, I'm on a sidetrack with habits because I love talking about those. You know, at the end of the day, these neurotransmitters do a lot of things in our body. They're these chemicals that our brain makes that bind to things and cause them to happen. Even like you look at a person who has like depression, right? Depression is not just somebody who is like in a funky mood or feeling sad all the time. That's totally not what depression is. Depression is actually where certain neurotransmitters in the brain, unfortunately, they just cannot bind. They're missing the binding sites. So what ends up happening is their brain is supposed to create some type of emotion, and it doesn't have to be good. It can be bad too. It could be anger or frustration or sadness or happiness or joy or comfort. It can be any one of your emotions. They're all controlled by these neurotransmitters. And in a depressed person's brain, those neurotransmitters can't bind very effectively. And that's why you'll hear somebody who's depressed, they'll describe it as feeling numb, not as feeling sad. Totally different situations. And that is why neurotransmitters are huge. They trigger everything inside of your body. So pivoting back to muscles, now that we have a really good understanding of what neurotransmitters are, because they're not in our notes today, the big one that we are going to talk about is called acetylcholine, or ACH, okay? Acetylcholine is the neurotransmitter responsible for triggering your muscle contraction. So this is what sets off your action potential. So you will have a ligand-gated sodium-potassium pump somewhere along this muscle cell. And what will happen is once a neuron gets a certain amount of electricity, it will release acetylcholine across this little gap right here. And that acetylcholine will bind to those sodium-potassium pumps. Those sodium-potassium pumps will get to work. They will start creating an action potential along your muscle, and your muscle will contract. How does it contract? Well, it releases calcium into the area. And remember, going back to here, once calcium comes in, troponin and tropomyosin move out of the way, and now actin can bind to myosin. sorry, other way around, myosin can bind to actin, and it creates that contraction. And then once that signal is done, the calcium is flushed out of there and the chaperones return back into place and your muscle gets ready for its next contraction.
I got a question.
Yeah, what's up?
When it comes to the bone, when you break it and you try to put it back together, can it be stronger than when you first had it, when it wasn't broken?
Yes.
Strong again or healthy again?
Yeah. So think of it like welding. You ever seen have you ever broken a piece of metal and seen like a welded piece of metal? What they do is they use a bunch of other, they fill in the gap and then they cover it with more metal, kind of like putting extra duct tape on something. Your bones will look kind of like that once they heal. Yeah, they can. It means that they're more resistant to breaking. Now, when we say stronger, we mean that section of the bone is more resistant to breaking. It doesn't mean that you can't break that bone again. You could always break it in a different part of the bone, which is often what happens. Because if you have a broken bone, if it was a really traumatic event, that's its own thing. But if you broke a bone and you didn't really do anything that hard, you could have like bone density issues. So yeah, your bone in that part will heal better, but it doesn't mean that you are immune to breaking that bone again. It'll travel, like I said, the action potential will travel down. That will release acetylcholine, and acetylcholine will trigger your action potential. And that triggering of an action potential releases calcium, which is Ca2, which is into the area. And calcium is an important part of muscle contractions because it is what binds to troponin and tropomyosin to allow for the contraction to occur. Okay. And then once that happens, you're left with nothing but a little bit of choline. That's the acetylcholine, right? That's the choline part, and it'll get recycled, and your body will just make some more of it, and send it right back to the area, and get you ready again. This is also a reason why we start to get a little bit tired. You kind of feel a little bit exhausted, and your body's like, oh, all right, cool it down, Tiger. You did a great job working out today. Let's maybe go take a nap. Because that's your nervous system being like, I'm out of juice, man. We got to relax. In a muscle contraction, right, your acetylcholine will degrade, producing choline, and then it'll get recycled and do it again. The end plate will depolarize, right? It'll switch those sodium and potassium ions, creating that membrane potential and releasing that calcium into this area. Don't worry about this name. But the area that's called is your sarcoplasmic reticulum. It's a really cool freaking name, but that's all of your SR. We refer to it as SR because who wants to say that all the time? But your SR is actually these tubes here. That's how that's all able to travel to your individual muscle cells because you've only got like a little connection with your neuron, right? There's only a neuron here. And so the electricity will run throughout the entire muscle cell. But you've got to be able to get like all that like calcium and stuff to like travel everywhere here, right? And it's only connected right there. So it's using this like network. I always think, again, like I said this yesterday, but I always think of like the SR as like subway tubes. It's how massive amounts of calcium are traveling from point A to point B and getting where they need to go. And so they'll travel to wherever it needs to get, and it'll get released into the fluid portion of the cell. That will allow, troponin to grab that calcium, and it will pull the tropomyosin with it. So now the proteins are out of the way, and your contraction is done. Your contraction is terminated. It's over. You did your contraction. By the way, we're going to get to this in just a second, but that is called a concentric contraction, is when your muscle is finished shortening. It's officially bunched up, right? Then what will happen is the ATP will detach because it burned up all of its energy in order to do that. And when the new ATP molecule binds to it, once it binds to it, that flushes all the calcium out. Once the calcium is gone, the chaperones return back into place and your muscle will relax. I kind of got ahead of it here. That's, yeah, it'll slide back and the sarcomere will lengthen. I got a little bit ahead of the slides on that one. So that is a very long process. Like I said, it's not described very well in this class. The big things we really want you to know in this class are your muscle fiber types. Because here's the thing, like I said, we're going to describe this much better next module, actually. But when you look at the different types of muscle fibers, you've got type 1 and you've got type 2. Type 1, they can do this all day. They've got so much endurance. They can just they got so much mitochondria. They are just producing energy all the time. But your type 2 fibers, they have a really hard time doing sustained continued contractions because they are going to burn up a ton of energy, and there's more muscle fibers. There's more of them. They're much larger in size. So they're going to be really good at creating these really big, really powerful contractions. But then after that, they're exhausted. You're going to need a lot of rest. And that's the difference, again, between like a power lifter and a marathoner. They're two very different types of athletes. Or if we want to use closer analogy, it's the difference between a sprinter and a marathoner. Both good, both different. All right, how we all feeling? I know that every time we do actin and myosin, people's brains start to melt. Are we feeling good, guys? We're going to pivot. We're going to pivot into the easy part of today. Yeah. All right. Here's the much gentler version, or the much gentler half of this PowerPoint. So getting all the way out of that, let's look at our types of contractions. So what happens, let's see, I got my little water bottle here. I don't know, this probably weighs about 2 pounds right now, I'd say, maybe 3. So what happens if I've got a muscle, my bicep here, and I generate 4 pounds of force, right? 3-pound bottle. If I generate 4 pounds of force, you know what's going to happen? I'm going to overcome that resistance. My muscle cells are going to fire in such a way that they're going to contract for more force than gravity can pull on this bottle. But if I generate an action potential and I generate a contraction that is weaker than 3 pounds of force, let's say I generate 1 pound of force, right? That's less than, the bottle is going to go down this way. It's not that I'm not contracting. I am contracting my muscle fibers. Make no mistake. There is a contraction that is occurring here, but it is a contraction for less force than is being put on the muscles themselves. So the muscle is relaxing back to its original full length. Still a contraction though. And that's something that people always forget. A lot of people think that contraction means to squeeze your muscle. That is true. That is one of 3 types of contractions. Okay. So of your muscle contractions, you have concentric contractions. I would say, by the way, guys, if you forget everything from today's lesson, this is the part I want you to remember. So concentric contractions occur when a muscle is shortening. Okay. So when your muscle cell does this action where it, is moving this towards here and this towards here, and your muscle cell gets shorter, that is a concentric contraction, right? You are overcoming a resistive force, right? It occurs when there is a need to overcome a load under gravity or stretching, like if it's a resistance band, or, swimming through a pool, if you're moving your arm through the water, right? All of those are concentric contractions, right? If I concentrically shorten my deltoid muscles, my arms will raise to the side. If I concentrically shorten my bicep muscle, my elbow will bend. If I concentrically shorten my pectoral muscles, my arms will come forward like this, right? So those are concentric contractions. Now, the exact opposite of that is when your muscle is relaxing back to full length, right? When your muscle is lengthening, that is called an eccentric contraction. That occurs when a muscle is lengthening. It occurs when a muscle is developing less tension than whatever resistive force there is. So if the resistive force is gravity, then you are descending, back down into the ground. If that resistive force is a resistance band, you are relaxing it back to its shortened position, right? So that is an eccentric contraction. And then there's the middle version, which is an isometric contraction. An isometric contraction occurs when your muscle is remaining at a constant length. So you contract your muscle fibers and then you leave them at that length. There is no change in the length of a muscle. It is generating an equal force to its resistance, right? So whenever we are lifting, right? If I'm doing, let's say, bicep, I always use bicep curls because they're so easy to show on camera. But if I do bicep curls, right, I go here and then I relax, right? Concentric. And if I held it here, that would be isometric. And if I lengthened it, that would be eccentric. So we actually use this whenever we're writing workouts. So I'm going to show you guys an example workout here. I'm just going to pull up, one random one here. Okay, that's blank. Good job past me. Can we not have blank ones, please? What am I doing in my life? There we go. Either it was actually blank or my computer is slow. So take a look here. This is sort of one of our NASA written out workouts. And take a look over here. If we were to do a single leg bicep curl, and we did it for 3 sets of 15 repetitions, and we do it at a 4-2-1 tempo, we are going to always write tempo in order from eccentric to isometric to concentric. It's always in that order. So if you've ever seen a workout and you've ever seen like tempo listed, it always goes eccentric is the first number, isometric is the middle number, concentric is the third number. They always write it in that order. Now that is kind of weird because you might be thinking like, wait a minute, if I grab a pair of dumbbells, the first thing that I do is concentric, right? Like the first lift. So wouldn't that be concentric, isometric, eccentric? No, it's never like that. We have a standardized way of writing this. And that way we never get confused. So it's always eccentric listed first. So when you see this tempo listed as 4-2-1, and I'm
sorry to interrupt you, teacher. What you say again? Can you tell me what is the order? What we would?
So when we write it out, we always list it as eccentric first, isometric second, concentric third. It always goes in that order. Even though like a bicep curl, the first part of it is the concentric part, that doesn't matter. Not when we write it down. Okay. So if I do a bicep curl at a 4-2-1 tempo, it's going to look like this. I'm going to lift for 1 because that's the concentric part. So I'm going to go 1. Then I'm going to hold the isometric part for 2. So I'm going to go 1, 2, and I'm going to lower it for the eccentric part for 4. 1, 2, 3, 4. And this is a single leg bicep curl, so it would look like this. I'd have my leg up, I'd have my toe pulled, and I would go 1, 1, 2, 1, 2, 3, 4. 1, 1, 2, 1, 2, 3, 4. And this is what my bicep curls are actually going to look like. Now, if I did this is really good if I'm a new client. If I'm new to working out, this tempo is great because look at how long it takes me to do every single rep. It takes about 7 seconds per repetition. So my brain is having so much time to learn how to do a bicep curl effectively. Like my brain is like really getting a chance to memorize good, proper, stabilized form, right? And my joints are learning how to like hold themselves and stabilize. My foot's doing all this work. I'm getting a little core activation here, right? This is great for a client who's in stability. But what about a client who is, and actually I do want to show you one other exercise. So let's look at a squat. A squat at a 4-2-1 tempo. So we went 1, 1, 2, 1, 2, 3, 4. A squat would look like this. 1, 2, 3, 4, 1, 2, 1. 1, 2, 3, 4, 1, 2, 1, right? Yeah, I started eccentric. This time I went in kind of the order that it was written in, right? So the order, the way it's written, does not indicate what you do first, like during the exercise. It indicates how long the eccentric is, how long the isometric is, and how long the concentric is. And depending on what lift you're doing, it's gonna, happen in whatever order it happens to happen in, if that made sense. So now I want to show you, what if I was a bodybuilder? What if my goal isn't to create stability? What if my goal is to create gains with a big Z, right? Well, then I'm going to do what's called a 2-0-2 tempo, okay? And a 2-0-2 tempo is going to look different. So now I'm not going to do, the single leg bicep curl. I'm not going for stability here. I want to be stable and I want to lift heavy, right? Because I'm only doing 8 reps here. So what I'm going to do is I'm going to do a 2-0-2 tempo, and we're doing a preacher curl, which I don't really have the ability to do here in my room. But imagine I were like sitting on a bench and my elbows were supported right here. So a 2-0-2 tempo would look like this: 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. Okay. See how
you need to be faster?
It is faster because it is 2 seconds eccentric, 0 seconds isometric, 2 seconds concentric. It doesn't necessarily have to be super, super fast, but we want to have an even distribution between concentric and eccentric, and we want to have no pausing in between the two. The reason for that is because NASM believes that if you do that, it will result in maximum lactic acid production. And we actually want to make lactic acid here because that's going to make our muscles burn the most. And if we burn our muscle fibers, your immune system will have to repair them more. It repairs them more, it will make them bigger. So this is a really good tempo if you're trying to grow your body. And the other tempo is really good if you're trying to create stability. Does that make sense?
Mm-hmm.
I told you we'd talk a little bit about lifting at some point. It's not all just biology. All right. And now lastly, what if I'm trying to get really strong? Right. Oh, hold on. I gotta show you this. I gotta show you a different one. So squats, by the way, it would look like this: 1, 2, 1, 2, 1, 2, 1, 2. Right. Now, what if I'm trying to get strong? What if I'm really trying to increase my strength? Well, now we're gonna list out our tempo like this: XXX, right? And that is an explosive tempo. That is super, super explosive, right? So you'll actually notice there's no bicep curl here. Oh, you'll also notice that I totally did not write it for the barbell squat. Sorry, that's gonna drive me crazy. 3 minutes. That was just for me. So, yeah, replace it. Yes. Sorry, my OCD is kicking in. So if we look at like you'll notice there's no bicep curl here. We really don't do single joint actions, biceps and triceps. We really don't apply them for like a lot of max strength workouts, or we really don't apply them very often for power workouts, because your biceps and triceps, these are like accessory muscles. These have a lot of endurance. They don't really respond that well to super heavy lifting. So if you want to grow your biceps, you'll do a little bit better at a medium rep range. And if you're doing a medium rep range, you wouldn't be lifting explosively. You lift a little bit lighter and you do higher reps. This tempo, this rep range works a lot better for compound lifts, things like a bench press, which is using my chest, deltoid, and triceps, right? It needs to use a lot of muscle together. So an XXX tempo would look very silly like this. And, hurts my elbows. So instead, let's look at a barbell squat. Let's say I had a really heavy barbell on my back, right? And I'm there and I'm going to do an XXX tempo, right? What I'm going to do is I'm going to get as much weight as possible onto that bar that I can do. I'm going to drop down explosively. I'm definitely not going to pause at the bottom. And I'm going to try to come up explosively. But it's really, really heavy. So what's probably going to happen is I'm not actually going to come up very explosively. What will probably actually happen is I'll come up like, holy crap, that's hard, right? So it'll look like this most likely. But I'll go, and then I'll lift and get it up, right? So it'll be down and then this, oh God, you know, like I'm barely getting it up. That's a really heavy barbell squat. I'm trying to do the explosive tempo, but I'm not quite able to pull it off probably, but that's okay. I'm still putting conscious effort into driving up out of that squat as fast as I can. If it's a deadlift, it would look like this, right? I'd drop it down and then I would lift that as heavy as possible, get it up explosively, and then drop it down. I really don't need to have this big long pause at the top, okay? Bench press, I'm gonna drop it down, I'm gonna drive it up, right? I'm trying to be explosive. But it'll be very heavy. So I probably won't look that explosive. Doesn't mean I'm not trying to be explosive, okay? And that's an XXX tempo. Could you do any other tempos? Yeah, there's some other ones out there. NASM's biggest competitor, for instance, they're called ACS, ACSM and NSCA. Those are two of NASM's like competitors. They are a big fan when it comes to hypertrophy of a 3-1-1 tempo. They believe that like doing it like this, 1, 1, 1, 2, 3. So it kind of looks like this. They're a big fan of that tempo for when it comes to hypertrophy. And they believe that like eccentric contractions cause more inflammation, and more inflammation will result in more muscle gains. NASM disagrees. I've goofed around with both. I think they're both really good. I actually don't dislike the 3-1-1 tempo. I'll use it whenever I'm trying to change things up. But in terms of you guys, do that once you graduate. For the next 9 months, the tempos that exist are 4-2-1, 2-0-2, and XXX. And once you're out of here, then you can do whatever you want. And 1-1-1. 1-1-1 is like your classic lifting tempo. You know, that's what you often see people doing in the gym, where they're just kind of going through the motions with a rhythm. So again, going back, what do concentric, eccentric, and isometric mean? Concentric is a shortening of a muscle. Your muscle is developing greater tension than a resistive force. It is winning the battle. Eccentric contractions is when your muscle is lengthening. It occurs when your muscle is generating less force than a resistance, which means that it is relaxing. And isometric contractions occur when your muscle is remaining at a constant length. It is creating equal tension to whatever resistive force you have. All right. Any questions on that, guys? Cool. All right, so lastly, let's look at our muscle fiber types, okay? So you've got two main categories of muscle fiber types. If we look at them, if we look at our type 1 versus our type 2 fibers, there we go. You'll see them, they look just a little bit different from each other. You will notice that these fibers are a little smaller, a little more tightly packed. They're also, there's a lot of red fibers here versus a lot of white fibers over here. And you'll sometimes, oh God, you'll sometimes hear these referred to as red versus white fibers. Red fibers have a lot of blood, therefore they get a lot of oxygen, therefore they're really good at endurance. And white fibers, they have a lot of myofilaments. They have a lot of like actin and myosin proteins. They're much larger, but they don't have as much oxygen delivery, nor do they have the fuel, or, you know, they don't also have like the tools necessary to use that much oxygen. So because of that, they don't really have very much endurance. But because of how much larger they are, they're much more powerful. They're much stronger. So if I'm training my slow twitch fibers, that 4-2-1 tempo is great. And if I'm training my type 2 fibers, that XXX tempo is great. So your type 1 fibers are what we refer to as slow twitch oxidative fibers. They are muscle fibers that have a well-developed respiratory system. They really breathe quite well, which ensures that they get a delivery of a lot of oxygen to the muscles. They're smaller in size, but they have way more mitochondria. They have way more hemoglobin. They have way more myoglobin, and we use them for long endurance type contractions. Okay. Type 1 fibers are generally associated with you doing cardio. Then we got our type 2 fibers. Now, until now, I've kept it kind of simple. We've only been type 1 and type 2. Type 2 is a little bit more complicated than that. There is type 2A and type 2X. And X stands for extreme, kind of. So type 2 fibers are your fast twitch fibers. These are muscle fibers that have a higher CA2 level, and they have more cellular volume. So they actually can store more calcium than your type 1 fibers, which, again, calcium is what flushes into the muscle fiber, moving the regulatory proteins, allowing myosin to grab the actin. So because of their much larger size, they can store more calcium, I'm not exactly the biggest guy in the world. I always joke around. What I always tell people is I'm the world's skinniest personal trainer, cardio athlete, you know what I mean? And so I don't have a huge storage of calcium ions. I don't have a large cellular volume in certain muscles, right? Someone like C.T. Fletcher or Arnold Schwarzenegger, like a bodybuilder, they're going to have a much larger cellular volume. They have a larger number of muscle fibers that their body has created. And then those muscle fibers have more cellular volume, which means they can create more energy using glycolysis. They've got more goo in their muscle cells, and that's great because that allows them to lift super, 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 super heavy. I should have said Eddie Hall. That's the strong guy. So your type 2 fibers in general, they are larger in size. They don't have as much less, they have less mitochondria, but they have more cellular volume. Remember, the cellular volume is the cytoplasm. That's the fluid of your cells. So they have more cell goo. They have lower hemoglobin, they have more, lower myoglobin, but they have way more actinin and way more myosin. And they are used for short-term powerful contractions. Now, there is a slight difference between true type 2 fibers, which are your type 2X fibers, and type 2A fibers. I want you guys to think of type 2A as like a stopgap between 1 and 2, right? They're fast twitch for sure, but they're fast twitch that still have a fairly good delivery of oxygen, which means they're still fairly good at having some endurance. When you think about a football player, or really any athlete, but football players in particular are a good example of this. These are the type of athletes who are very explosive, but they've also got a pretty good amount of endurance. I play a lot of, you guys don't know this, and I talk about it all the time. Noel and Nicholas are sick of hearing it, but my favorite sport is Ultimate Frisbee. I play a lot of Ultimate. I play in leagues across the city, and I play in a pickup game that I've played in for 10 years. If any of you guys ever want to come, you are more than welcome, by the way. Love having students come. It's a blast. But it's my favorite sport because it's like soccer. It's all the cardio of soccer with all the elegance of getting into an end zone like football. And so it's in my opinion, it's the world's greatest sport. And we were playing just before winter break and stuff. And we saw this guy working out in the park, and he was tearing it up. I mean, this guy was super fast. And it turns out he is in the Canadian Football League. And he was like, "Can I play?" And we were like, "Yeah, come on in." And he is explosive. He is fast, but he still has a fairly good amount of endurance. But if you want to beat that guy, you just got to jog like crazy for about half the game. And you have to accept that you are probably not going to score for a good while. But then after a while, eventually, if you do enough jogging, he runs out of endurance. And then you can, kind of keep playing at a slow pace. And so he's a very good example of a type 2A athlete. He has a fairly good amount of endurance, and he's still very explosive, but he doesn't have as much endurance as a type 1 situation. But he is extremely fast, right? He's extremely explosive. So that's when it comes to athletics, when it comes to sports, what you really do want to have is a lot of type 2A fibers. We don't need a football player to be as strong as Eddie Hall, right? Eddie Hall is a very famous power lifter, by the way, for those of you who don't know. Or the Mountain, Thor Bjornsson. He's another very famous power lifter. Those guys are great, and they're extremely strong. They are, you know, the strongest men on the planet, literally. They have won the competition. They both have like the records for the heaviest deadlift on the planet. That is example of somebody who is very much true type 2. But like if they had to like jog a mile, you know, they would do it, but they're going to get exhausted fairly easily. Whereas like a football player, they may not be as strong as Eddie Hall, but they can play an entire game. Or like look at LeBron, right? Like he can play an entire game. He's still very, very, very explosive. And even with his long freaking limbs, I mean, when I think about like how strong basketball players in relation to how long they are, it always blows me away. So in sports, we typically want to have type 2A fiber. Most athletics rely on type 2A fibers. These are fast twitch, but they're still slightly what we call oxidative glycolytic fibers. They really rely on that glycolysis metabolism to give you some endurance, but not quite a ton. Then we have type 2X fibers. And your type 2X fibers are your true fast twitch fibers. They are extremely explosive, they are extremely strong, and they have very little endurance. But that's okay because they're super explosive. These are your fast twitch, simply just glycolytic fibers. Type 2B or type 2X. It says 2B in this formula here. It will say 2X in your NASM test. On your actual NASM textbook, it'll be 2X. That's where we start to, every now and then we run into these little disagreements. They really do not have very much blood flow. They don't need it. And they're very, very good at relying on actually a metabolism that we haven't really talked about yet, but what is called your phosphagen system. They actually very much rely on this energy system that exists before glycolysis even takes place. And so that's going to be like 1 to 5 reps. So this would be like 1 to 5 reps. This would be like 4 to maybe 10 reps. And this would be like, you know, infinite reps up to 20, you know. And that's what we're looking at. So when it comes to modifying your muscles, you know, endurance type training is going to trigger the conversion of type 2X fibers into type 2A fibers. So if you are an athlete, if you are a football player, it's actually a very good idea to do what is called periodization. Nicholas, you and Noel, you guys know what I'm talking about when I say periodization. Periodization is how we as trainers go through different training cycles. So if I'm trying to make an athlete be very explosive, I'm going to work on making them explosive for maybe 4 to 8 weeks. We're going to do a bunch of heavy lifting with long rest periods. And that's going to develop a whole bunch of type 2X muscle fibers. Then I'm going to start lifting slightly lighter, but I'm going to still keep it relatively heavy. And I'm going to try to convert those 2X fibers. I'm going to start to give them just a little bit of endurance and turn those into type 2A fibers. So we're going to kind of fire up the cardio right before the season starts. So I get them real strong. I give them a little bit of endurance right before the season starts. And then in the mid-season, you know, I'm going to keep them kind of honestly just doing some gentle workouts because mid-season, your priority is to make sure your athlete doesn't get injured. It's not about, like, LeBron's not trying to improve his 1 rep max mid-season. He's working on getting to the playoffs, you know? And that's true of any athlete. But that's where we start to go through different cycles, right? We cycle through like high intensity to low intensity to high, and you know, we do, it's a big circle. And then if you were to like continue just doing cardio and cardio and cardio and cardio, well, your body's going to go, holy crap, we need to produce energy a lot. So it's going to start converting those maybe to type 1 fibers, and it'll start getting more blood flow in there. More, your red blood cells will have more hemoglobin. You know, those are the adaptations that we start to see. And so a high percentage of type 1 fibers is necessary for endurance. But Type 1 fibers do tend to be a little bit of a genetics thing. This is where we start to see just a little bit of genetics come into play. Generally, you, based on your genes, are going to have a greater predisposition for either type 1 or type 2 fibers. If you look at everybody in my family, we're all, long, skinny people. I come from a tribe of people that, when foreign invaders would show up, we'd be like deuces and run that way, and they would never catch us. Whereas like other people come from a much stronger, a more of a type 2 genetic, predisposition. Everybody's going to be a little bit different. You're going to have a predisposition for one or the other. However, I want to be very, very clear here. That does not mean that you cannot train to develop your body however you want it to develop. Your body form follows function. Your body will adapt specifically to whatever stress you place upon it. You want to get back, if you're somebody who's like, oh, I've just never been able to run. Well, if you start and you're consistent about it, you'll get good at it. I promise. And if you're like, oh, I've never been able to gain muscle. Well, if you do the things necessary to gain muscle, it will happen. Yeah, some people are going to have an easier road than you are, but that doesn't mean it's impossible. Just like some people got to get a $1 million loan from their parents when they were, younger, and start an empire that eventually led them to be the president. And other people, they don't have that much. No statements. So that is our lesson. Doesn't mean you can't get there. Any questions on today's topics? How are you feeling, guys? Confident?
Excellent.
Great. Yeah.
Cool.
Also,
Really good, really good.
Awesome. Yeah, what's up?
yeah, well, like they say, this is the very too much definitions of all these concepts.
Yeah. Work on memory. Yeah. Get those, the actin, the myosin, troponin, tropomyosin, get that down. I would definitely memorize those, and then work on your muscle fiber types. Everything in the notes, like I said, the PowerPoint is the PowerPoint's a dictionary. The notes are the terms I want you to know. Okay.
Okay. So the notes always want to be the base of to a study.
Yeah, the notes are, I wrote those notes, I pulled the stuff out of the PowerPoint that is most relevant. I try not to put too much in the notes. Sometimes I'll put stuff in the notes that is from other classes. So it's important, even if you're not tested on it in this class, it's still something you'll see later. So every now and then the notes will have more than you need, but for the most part, they are there to act as a filter. But guys, you made it through your first week. We've got our first meeting in the park on Monday. We are going to meet at 9 a.m. instead of 8:30, or at 6 p.m. instead of 5:30. That way, if anybody needs an extra, if your commute is long or whatever, you got a little bit of extra time to get there. I will be sending out the address to the park. I'm going to say this on the call right now because this happens every single time. The address that I'm giving you is the address I want you to go to. It is the address for the parking lot. So go there and you will find us. Inevitably what happens is one person will always just be like, oh, it's called Van Nuys Park. And then they punch that in and then it will take you to the wrong parking lot. Don't do that. Go to the address that I put in the email.
Okay.
So I will have details for you there. Everybody's going to bring their mask. Generally, we're outdoors, so we'll separate so that you can take it off, and we'll be outdoors. So, we'll be 6 feet apart and stuff, and it'll be okay. I don't need you to sprint around and run with your mask on or anything like that. It's going to be a little bit of cardio, and we're going to talk about muscular contractions. So we're going to practice eccentric versus isometric, just so you can kind of get those in your bones. We're going to do those plenty of other times as well. But it's, it is optional. just so you guys are aware. I know not everybody can make it, or some people might live with someone who is immunocompromised, and you're trying to limit your interactions with people. If you cannot make it, just let me know. Just because like sometimes what happens, like everybody will go to the morning session, and then like I'll show up for the evening session, and then nobody will, nobody will be there. So just let me know if you're not going to make it. Like I said, it's optional. If you can't make it, you're not necessarily missing anything that's going to help you pass your tests or anything. It's just really a chance for us to like practice stuff. We will have no lecture on Monday because I'm going to be in the park, but I will be putting out an email, and there will be about a 30-minute video that is meant to act as a summary of everything we talked about so far. So it's a video that I recorded in the past for the first time we ever taught this class on Zoom. It's just a 30-minute video where I am going through. When I created a special notes sheet, it's like, how the notes filter the PowerPoint. Well, this note sheet that I get out on Monday it's filtering everything that's on the test, and so. It's just something for me to talk about, and it'll just be me talking for about 20 to 30 minutes, giving you the most important like bare pieces that are going to be in the test. And then the test is going to be 30 questions. It will, Nicholas is like, I've heard this amount. It's going to be 30 questions, and it will, 15 of those questions will be questions you guys have already seen before. You will have seen them in homework 1 and homework 2. So there'll be 15 new questions, but they will only be from PowerPoints 1 through 5. So the 15 new questions will still be familiar. It's just like, maybe homework 1 was like, let's see here. What is the acid in your, I don't know any of the questions off the top of my head, but what is the acid that's in your stomach called? And you would say hydrochloric acid. And so you had that on homework 1, and it shows up in the test as well, right? But then maybe another question that's on the test that wasn't in your homework is, what is the name of the first part of your small intestine? And that's your duodenum, right? It's from the same PowerPoint. It's from the same lesson. It's just not something you've necessarily seen before.
Okay, I want to understand. You say the first 15 questions want to be like
So not the first 15. They'll be jumbled.
Okay.
Yeah, it's not going to go in order. They'll be jumbled up. But 15 of them you have already seen. It'll probably, if I'm assuming right, it'll probably be 7 questions from homework 1. And 8 questions from homework 2 will be in the midterm. And then the other 15 will be from the same topics, but you haven't seen those questions yet.
Yeah, but want to be in the note, or the other questions want to be from the notes or something. Definitely. Like that?
Yeah, it won't be, it'll not be, it will always be something I've already taught you. Yeah, it will be in your notes. It'll be in the PowerPoint. Yeah, you'll know it. You'll know it. Honestly,
we want to need to study both the notes and the PowerPoint too?
I would. It's helpful. Yeah. Study your homeworks, study your notes. I would say the notes over the PowerPoints, if I'm being honest.
Because like you were saying, in the PowerPoint, sometimes they have too much information. So.
Yeah. So I'd probably put more emphasis on the PowerPoints than I would on the notes. Oh, blah, 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 blah. I said that backwards. I would put more emphasis on the notes than the PowerPoint. Sorry.
No, you scare me. You scare me. I say, oh, wow. When I read the one.
Sorry. I'm worn out. Yeah. So, yeah, put more emphasis on the notes. And watch the video. Watch the video I put up on Monday. Those review videos are super.
Okay, perfect.
Hey, Brad.
What's up, Noah?
On the PowerPoints, is the questions on the PowerPoints the same as the NASM is going to be on the NASM test?
Close. Yeah, we always get this question, right? The PowerPoints were written by NASM, right? So that means that, it's the same staff of people. So it's really similar, but no, they're not the same questions. NASM does not release their questions. They keep those really closely guarded, but in the same way that the questions in your homework are similar to the questions that are on the test, the questions in the Sochi program are similar to the questions on the real test.
All right.
And well, I told her yesterday I kind of go to the park because I have the coffee and I need to go on with more.
Actually, that's a good point. Anybody else as well? I should probably put that in the email, and I always forget to say that. Anybody else who has tested positive for COVID within like the last 14 days? Yeah, we're not gonna have you come. You know, we want to make sure nobody's got it. You know, but that's why we're all gonna be masked up. But that's okay. Sorry, we're gonna get you on the next one.
Yeah,
no
and I want to ask you too if you can make a video for what you want to say on Monday.
It's very hard for me to film this. I don't have like a ability to kind of film while I'm out there. Unfortunately, I just don't have the equipment, or I don't even have the knowledge. I don't know how to film stuff, to be honest. You know, like you wouldn't at some point I'd probably be too far away from the microphone. I wouldn't be able to be in frame or whatever. Unfortunately, I can't record the in-person meetings.
Okay.
What's that?
No, it's okay. Thank you.
I wish I could. If I could, I'd start a YouTube channel. Andres, how you feeling?
I'm good.
Yeah, Kenny?
All good. All good.
Awesome. All right, ladies and gents, I will see you guys on Monday. And if I don't, I'll see you on Tuesday. Have a great weekend.
Alrighty. You too.
Bye, brother.
And one question more. When gonna be the